The suprachiasmatic nucleus, often just referred to as the SCN, is located in the hypothalamus and is the master circadian clock, organizing our 24-hour rhythm, including sleep and wake. A series of studies in the 80s and 90s showed that lesions in this nucleus in animals resulted in a total loss of circadian rhythms. You might say these animals suffered severe jet lag. However, the rhythms were completely recovered by transplanting tissue of this nucleus from another animal. The SCN is called the master circadian clock because most of the cells in your body also contain circadian clocks. Some of these peripheral clocks independently set the clocks of your liver, gut, and other body organs, but they need the master circadian clock to all be synchronized together. We mentioned before that there is a direct track between the retina in the eyes and the suprachiasmatic nucleus in the hypothalamus. The retinas detect light, and as you might recall, natural light is the #1 factor in the setting and resetting of the master circadian clock. First, the master clock must be reset in order for it to then synchronize all of your peripheral body clocks. When the retina detects dark, the absence of light, the SCN triggers the pineal gland to start making melatonin from serotonin. This melatonin, combined with the adenosine you have built up during the day, cause you to get sleepy and eventually build up to flip what is called the sleep switch. The ventrolateral preoptic nucleus, and this is often simply referred to as the VLPO. VLPO. The VLPO is another set of cells in the hypothalamus that play a critical role in producing sleep. McGlinty and Simusiak in the year of 2000 discovered its major role and thus named it the sleep on/off switch, oftentimes just referred to as the sleep switch. VLPO cells become active around the onset of sleep, and their level of activity increases with the depth of sleep. The VLPO sends inhibiting messages to the arousal systems, including those that use orexin, histamine, glutamate, and serotonin to maintain wakefulness. As these levels decrease, GABA levels increase and continue to increase through non-REM sleep. When non-REM sleep builds, two things happen simultaneously. Remember the basal forebrain? The basal forebrain uses adenosine to inhibit its release of acetylcholine, which in turn inhibits activity in the forebrain and increases in GABA levels. The cells in the basal forebrain that perform this function are one of many examples of areas of the brain that are more active in sleep than when we are awake. Meanwhile, the reticular formation in the brainstem still sends outside sensory information up to the thalamus. But remember the role of the thalamus as a gatekeeper for sensory information? During non-REM sleep, the thalamus closes that gate, thus inhibiting this information from going on to your cerebral cortex for interpretation. Remember that GABA levels are increasing. Thus, we now see the production of sleep spindles and K complexes during stage 2. So non-REM sleep is largely a product of the higher brain, though it could not happen without the lower brain doing its very critical thing of keeping you alive, right? Sensory information is also blocked by the thalamus during REM sleep, but there is less to block than there was in non-REM sleep because more inhibition takes place at the sensory receptors, spinal cord, and brainstem before even reaching the thalamus. While the thalamus helps to suppress the cerebral cortex during non-REM sleep, it helps keep it active during REM sleep. The brain is highly active during REM sleep, as high as it is when you are awake, and many parts of it even more active, partially due to the forebrain resuming its release of acetylcholine. Recall that's the main chemical in wake and is inhibited in non-REM sleep. However, except for automatic functions like your breathing and heart beating, for example, the active brain is nearly isolated from the inactive body in REM sleep. Very limited sensory information is making its way into the brain, and commands from the brain to the voluntary muscles of the body are blocked at the brain stem by glycine and GABA working in concert, producing what is known as REM muscle paralysis. Finally, there are three specific groups of cells in the brainstem that are maximally active during REM sleep, collectively called REM-on cells. Other cells in other areas of the brainstem are nearly silent during REM sleep, and thus called REM-off cells. Now, don't get these mixed up with the sleep switch in the hypothalamus, right? As their names imply, REM-on cells are active during REM sleep, but REM-off cells are active during non-REM sleep. So while non-REM sleep is largely a product of the upper brain, REM sleep is largely a product of the lower brain, particularly in the pons and midbrain parts of the brainstem. Thus, REM sleep is sometimes called primitive sleep. In fact, if you remove the cerebral cortex of the human brain, REM sleep would continue, but what we know as non-REM sleep could not.